No, you can redo the,
Okay, but have everyone joined?
yeah, joining. Yeah, don't worry.
So fine. So our apologies for the technical problem. So Dr. Damre is an assistant professor at University of Goa University and has a PhD in marine sciences and a background in microbiology. Her career includes roles as a research associate at CSIR, National Institute of Oceanography, and a postdoctoral fellow in Germany. She has managed a DST project under Young Scientist Program and has a history of academic excellence demonstrated by awards and fellowships. Dr. Damre's research on Thraustochytrids and Aphaenochytrids is well regarded and has published extensively, contributing to the understanding of marine microbial ecosystems. She actively engages in scientific communities and has delivered talks at various national and international platforms. We welcome you, Dr. Damre, for this session of International Seminar on Marine Fungi. And the floor is all yours. It's great to have you. Please go ahead with your talk.
Thank you very much, Dr. Sharma. I'll now share my screen. Can you all see my slides?
Yes, it's visible. If you can put it on.
Yeah,
Thank you so much.
Okay, I have now put it on slideshow. Are my slides moving?
It's moving. It's good.
Okay,
Great.
okay, fine. Yeah. Okay, so shall I start?
Yes, please. Yes.
Yeah. So good afternoon, everyone. My topic for today is Algae-Thraustochytrid associations in the marine environment. So this is the outline of my talk. I'll be introducing you all to the field of Thraustochytrids, and I'll let you all know the biotechnological importance of this group of organisms, the habitats that they are found in, the association of Thraustochytrids with other organisms in the marine environment, their associations with the microalgae and the macroalgae, and finally I'll end with the ecological role. So to begin with, what are Thraustochytrids? This tongue-twisting names, I saw you took really get effort to pronounce these names. So this group of organisms, they are also called as Traminopelon fungi because these organisms, earlier they were grouped under kingdom fungi, and later on, from the biochemical characteristics of the cell wall, the ultrastructural features and molecular studies, it was found that they no longer belong to kingdom fungi, but they fall into the kingdom Straminopela, which is under the SAR group in the eukaryotic tree of life. So fungi belong to Opisthokonta, and Thraustochytrids are in the kingdom Straminophiles, and therefore they are also called as Straminopelon fungi or fungal-like organisms. This term Straminophiles was coined by Dr. D.J. Patterson based on the tuberoclastate mitochondria present in these organisms. So here in the SAR group, along with Thraustochytrids, you have the diatoms, foraminifera, which also possess these characteristics. And the main characteristic is the zoospores that they produce. They are biflagellate, and the anterior flagellum, the tinsel type flagellum, and the posterior, the whiplash flagellum. Now they have osmoheterotrophic mode of nutrition, which is similar to that of bacteria and fungi. And the characteristic feature is they produce ectoplasmic net elements. Now, what are these ectoplasmic net elements? This picture that you see here at the background, these are the cells of Thraustochytrids. And through the plasma membrane, they produce some extensions. Can you see my cursor here? These are the cells, and these fine filaments that you see extending from the cells. These are the ectoplasmic net elements, which are the extensions of the plasma membrane. And it is with the help of these ectoplasmic net elements that these organisms, Thraustochytrids, attach to any substrate and they release extracellular enzymes and absorb nutrition. The cell wall of these organisms are made up of circular scales and they consist of sulfated polysaccharides and proteins. Now coming to the biotechnological importance of these organisms. So why is it important to study them? The main characteristic feature is they produce large amounts of polyunsaturated fatty acids, especially the omega-3 docosahexaenoic acid. And many of you must be aware of this importance of these PUFAs, polysaturated fatty acids, for us in humans, that they play an important role in the development of the brain, the heart, the eyes of us, and they are important for the even for reproduction and like the prevention of certain diseases like heart diseases, hypertension, in arthritis, cancer. So therefore, since these organisms produce large amounts of polysaturated fatty acids, these are extracted, the fatty acids from these organisms are being extracted or exploited commercially so that they can be made available to us. These are the cells of Thraustochytrids. When they are stained with the dye called Nile red, the lipid globules, the fatty acids which are produced in them, they will be fluorescing like this. And the photograph that you see here are the colonies of Thraustochytrids. And you can see that some of them are colored. So that is another characteristic of this group of organisms, that some of them produce carotenoids, which also have physiological role in us, like the activation of immune system for prevention of cancer and so on. Then another feature is that they produce squalene, which is structurally similar to beta carotene. Now here in this photograph, you can see that there are other sources of squalene as well. Now squalene is also important or biotechnologically important because it is required for the maintenance of our brain. Then it is also used for protection against UV or ionizing radiations, and it acts as a sink for highly lipophilic xenobiotics. So therefore, squalene plays a role in pharmaceutical industry. Then these organisms also produce saturated fatty acids, which can be converted into biodiesel. So this is a picture from a paper by Soydin et al. 2020, wherein they have shown how these organisms, when isolated from an environment onto the plate, can be cultivated in liquid broth, and with media optimization, they can be cultivated at large levels, and the fatty acids from these organisms can be extracted to produce biodiesel. So this picture is an overview of all those different types of molecules which these organisms can produce, and therefore these are important or biotechnologically important. For example, the first one is the omega-3 docosahexaenoic acid, which are used as dietary supplements also. Then saturated fatty acids for the biodiesel, carotenoids again as dietary supplements and in aquaculture, squalene for cosmetic applications, and finally the cellular mass, the biomass, can be used in aquaculture as animal feed. Now coming to the habitats where these organisms are found. Now, these are exclusively marine organisms, marine in nature. They are found in large abundance in mangrove ecosystem. So, mangrove ecosystem is a very, very good source for isolating Thraustochytrids. They are found in the water, seawater, the sediments, the detritus of the mangroves. They are also isolated from drifting algae, such as the sargassum or the brown algae, fucus, another type of algae. Then they are also found in large numbers in the seagrass beds. They are found to be associated with the salt strings. Now, salt, these are the zooplankton, which can be a single organism. And here, what you see here is a string, that is, many salps come together and they form such huge strings. And these Thraustochytrids have been isolated from the fickle pellets of these salps. Now, these are to show you the difference between the the size of the fickle pellet of the salps and the other zooplankton, like the copepods. Like the copepod pellets are so tiny, salt pellets are so big and Thraustochytrids have been isolated from these fecal pellets. Now, how are they associated with other organisms? For example, the mollusks or the sea bivalves, they were found in Haliotis, in Mercenaria, then in flatworms, also along with octopus, the squids, the nudibranchs, and I told you they are found in large abundance in the seagrass beds. They are also isolated from the seagrass called Zostera marina, and most of these associations, the Thraustochytrids have found to be parasitic. So the Thraustochytrid, especially known as Labyrinthula zosterae, is parasitic to Zostera marina. Then there are associations with the zooplankton. Now, zooplankton, this was during my doctoral studies. I had developed a molecular probe using the ITS, the internal transcript spacer regions of these organisms. And I developed a molecular probe which helped me to detect the presence of these organisms in situ in the zooplankton. And I could make out that these were found to be present in the gut of this zooplankton. So, and it was not just one specimen, they were found in many, and a lot along with the Chitognas. Chitognas are the ferocious predators in in the marine ecosystem, what to call them, and the Chitognas were always found to be present or having this Thraustochytis in association with them. So it was kind of a symbiotic association. And Thraustochytrids are also eaten up by the zooplankton, so they fall prey to the zooplankton for the same reason, maybe because these organisms produce pufas, and zooplankton need pufas for their growth and development, but they cannot produce pufas on their own. So they have to depend on a dietary supplement, and the Thraustochytrids, since they are producing pufas, the zooplankton will consume them and get their nutrition. Now, coming to their association with microalgae. Now, what are microalgae? Now, microalgae are anything which are photosynthetic. They are producing food by fixing carbon dioxide, and they are generating the organic matter. So they are the basis of the food chain. For example, the phytoplankton, the diatoms. We have the cyanobacteria also. So all these, there are different types of macroalgae, and they are important because they influence the global climate and the atmospheric carbon dioxide concentration. Every second breath that we take is due to this phytoplankton, because they are producing oxygen by generating or by fixing carbon dioxide and generating food. Now, Thraustochytrids have also been found to be associated with microalgae, and how? This is what I will be telling you all today. Now, here we will see where phytoplankton are in this marine food web. So this is the aquatic food web, where we have the phytoplankton, which are the basis of the food chain. Phytoplankton being eaten up by the zooplankton by the fish, small fish, and then small fish by the big fish. Now, these organisms, they produce dissolved organic matter and particulate organic matter during their as a part of the living cells or during the metabolism. And it is this DOC or DOM and POM which is being utilized by the microbes, and they form the microbial loop. They will take up this dissolved matter, and the bacteria, here you can see the bacteria shown here. The bacteria will absorb this dissolved matter and they will form the biomass. And the Bacteriovorous protist will eat them, and thus whatever matter is lost in the form of DOC will again come back into the aquatic food web through the microbial loop. Now, where do Thraustochytrids come into picture over here? Now, Thraustochytrids, as I told you earlier, have osmoheterotrophic mode of nutrition similar to that of bacteria. They also are part of this microbial loop here. And so they will be present along with the bacteria in this microbial loop. Plus, because they will absorb the nutrients by releasing enzymes, they will absorb nutrition from the substrates, and they are in turn eaten up by the zooplankton. So they are passing on the nutrition to the higher level. So they are an important part in the microbial loop. They are not just the decomposers or the saprophytes, but they are also prey to the zooplankton. So therefore, you can see here, this picture shows clearly how the POM or the DOM released by different organisms. For example, the photosynthetic eukaryotes, the mixotrophic eukaryotes, for example, the dinoflagellates, and the phototrophic prokaryotes like the cyanobacteria. These all, when they release them, they are taken up by the heterotrophic bacteria and our Thraustochytrids, and they will form, therefore the Thraustochytrids will form a part of the POM, and as the particle size will increase, it will form a larger and larger aggregate, which will increase in the weight or the density, and therefore it will sink to the bottom of the ocean, and it will be formed, it will be at the bed of the ocean. So with this background, what kind of studies I did was I collected seawater from various depths in the water column in the Indian Ocean, and I tried to see the abundance of this group of organisms, the Thraustochytrids and bacteria, and I tried to compare them. And also I checked the abundance or the concentration of transparent exopolymeric particles, which are nothing but the source, or they are the binding agents wherein the different ions or substrates will come and attach to them because of their sticky nature, and they will form the aggregate. So this, when abundance of Thraustochytrids and depths was checked at different depths in the water column, I could see in some stations there was a strong positive correlation between the two, and whereas in some, the concentration of Thraustochytrids was higher at certain locations just next to the peak of the Thraustochytrids. So if they are associated with these steps, are the steps a good source or good substrate for growth of these Thraustochytrids, and whether Thraustochytrids will help in degradation of these steps is something that we didn't know. So therefore, I tried to generate these steps and aggregates in the lab in roller table experiments. So this is the traditional roller table designed by Shanks and Edmondson, and this was the roller table that we designed at NIO in the workshop. And we tried to get or develop aggregates inside this using different substrates. So the first in an experiment, I grew diatom Ketosiros in F/2 medium, and I collected the culture filtrate. Now diatom will also release DOM, and that is the EPS, which is the good source of TEPs. So I used this as a source to inoculate in those roller table bottles. So it will have a good source of TEPs originating from diatoms. Then in another experiment, I took bacteria which are associated with these diatoms, grew them in zobel broth, and then extracted the supernatant. From the supernatant, I extracted the EPS by precipitation and used this EPS as a source of TEPs in one of these bottles. And then I carried out this experiment to check whether Thraustochytrids could grow in the presence of the TEPs produced by diatoms and the TEPs produced by bacteria, and which of these are good source of Thraustochytrids. And along with Thraustochytrids, I also enumerated bacteria and TEPs. And I found that the numbers of Thraustochytrids increased in the presence of diatom polysaccharides, but in the presence of bacterial polysaccharides, though they grew, it was not as much high as in the presence of diatom polysaccharides. And the bacteria numbers increased steadily along with that of the Thraustochytrids. And in case of transparent exopolymeric particles, their numbers were always increasing during both the conditions, in the presence of diatom taps also as well as the bacterial taps. The aggregates which were generated in this roller table experiments were then picked up and they were stained with acryflavine stain, which is by acryflavine direct detection technique. Now, this stain is a peculiar stain for staining Thraustochytrids, and because when Thraustochytrids are stained with this acryflavine, the inner contents of these cells will be seen as fluorescent green or yellow, and the cell wall will be stained as red. Whereas when other photosynthetic organisms will not be taking up this acryflavine stain, but the heterotrophic organisms, the protists, will take up this stain, but their inner cellular contents and the cell wall will not be differentiated. You will see them as red patches only. Then these aggregates were also stained with chalcofluor stain to see whether they have fungi present in these aggregates. And I could see that wherever, in whichever experiment I've added antibiotic to stop the growth of or to control the growth of bacteria, there along with Thraustochytrids, there were fungi also seen in the aggregates. So aggregates formed in the water simulated by the addition of antibiotics were favorable for growth of not only Thraustochytrids but also fungi. Now, coming to the diatoms, I worked this in collaboration with one of my colleagues who was working with Cylindrotheca clostridium, Skeletonema, and Thalassiosira. So what we did was we saw how the growth cycle of this particular phytoplankton is, and then we collected the extracts from these diatoms and we checked the fatty acid levels of these different diatoms. And we where fatty acid extraction was done by GC, by FAMES analysis, and each of these showed a varying pattern. Then these fatty acids which were extracted were used to see whether they have an influence on Thraustochytrid growth or not. So the Thraustochytrids were grown in this extracts of diatoms, the extracts which are collected at different life stages, that is the growth phase, the exponential phase, and the stationary phase. And we could see that they exhibited zone of inhibition against the Thraustochytrids. These on the x-axis, these are the Thraustochytrid cultures. 4 different cultures were tested with this extracts of 3 different diatoms. And as you can see here, this particular diatom showed a very good zone of inhibition along with this one. So the lipid which were extracted from the diatoms, especially during their late stationary phases, were more inhibitory to the growth of Thraustochytrid cells. And that too, only a few types of Thraustochytrids, not all were of this 4. 3 of them were inhibited by the diatom extracts. Now, for example, Schizochytrium species, it was found to be the most resistant to the lipid extracts of the diatoms. Then Oblongichytrium was found to be, it was like the stationary phase extracts were inhibitory to Oblongichytrium, but the logarithmic phase extracts were not at all, they had no effect on Oblongichytrium. So it could grow in the presence of the extracts which are extracted during the logarithmic phase. And as compared to Schizochytrium and Oblongichytrium, Parietichytrium species was found to be the most sensitive. That means all the extracts, the logarithmic phase extracts and the stationary phase extracts of the diatoms were inhibitory to the growth of Parietichytrium. So the diversity of allelopathic interactions between Thraustochytrids and diatoms in the marine ecosystem was studied by this, and the pivotal role of diatom lipids during such interactions across different trophic levels could be seen. Now coming to the association of Thraustochytrids with the macroalgae. So macroalgae, which are seen at the intertidal regions on the beaches, especially on the rocky shores. Here are a few examples during our study. You can see the grubs, sargassum growing on these rocky beaches. This is Stochospermum, this is Spadina. Now, when these organisms were, I tried to isolate these organisms along with, from this macroalgae. Now, two different studies were done. One was the algae which were attached to the rocks, and the other one was the algae which were found to be drifting, the drifting algae, which were not attached to the rocks. So, when these Thraustochytrids were isolated from the drifting algae, and they were then checked whether they could tolerate the different varying concentrations of heavy metals, and the SCM studies were done to see how the cell responded to these different concentrations of heavy metals. So these are the scanning electron micrograph pictures of this Thraustochytrids in the presence as well as absence of this heavy metal. The first one is when they were, this was the control, that is without any metal, and this is in the presence of metals. You can see that they produced high amounts of EPS in the presence of metals. This could be because when the EPS is produced, it will be covering the entire cell and protecting the cell from the harmful effect of the metal. And secondly, some of them produced spores to the cell wall of this Thraustochytrids, to the Thraustochytrid cell wall. So in this study, we could see that Thraustochytrids were commonly associated with the sargassum, and some of the cells, some of the Thraustochytrid species could tolerate this heavy metals, and hence they can be used for bioremediation purpose. Then coming to the rock-attached microalgae from the intertidal zone. Now this study was done seasonally. Every month, the seaweeds or the macroalgae were sampled from the rocky intertidal shore of Goa. And in the month of August, these were the different macroalgae seen. You can see there were all green macroalgae found during monsoon, during the month, in the month of August. In the month of September also, which is a monsoon, there were all green algae. Then in October, the diversity of macroalgae increased. We had green algae, and along with that, the red and brown algae also. Then in November, this was the diversity of the macroalgae present in the same shore of Anjuna Beach, Goa. These were the different macroalgae found during December, and these were during January. So some of these species were seen during all the time. Like for example, sargassum was seen over here also in October also. Padina species was also found all the time. Then this was the diversity of macroalgae found in the month of February. This was during March. The diversity of macroalgae was very high in the month of October and March. Now in this study, the macroalgae which were collected from the intertidal shore, they were brought to the lab in cold conditions. They were washed about 5 to 6 times using sterile seawater, and they were cut into fine pieces, and then pine pollen baiting was done for isolation of Thraustochytrids. Now, pine pollen is the pollen grains of pine are used as a bait. Here, this is a picture of the pine pollen. Now, these pollen are used as a bait, or they are used as a substrate for isolation of Thraustochytrids, because Thraustochytrids will attach, the cells will attach to this pine pollen, and they will, through the ectoplasmic net elements, they will secrete enzymes and they will grow on this. And then the spine pollen can be picked and streaked on the agar plate. Now the agar plate has to be supplemented with antibiotics so that the bacteria will not grow and you will only get Thraustochytrid cell. And then the Thraustochytrid colonies are then picked up and then purified. And after that, the pure culture which is obtained is can be grown in liquid broth. The morphological studies can be done, their cellular character, the life cycle studies can be done for identification of these organisms. So in this study, what I did was the different types of macroalgae which were collected, the green macroalgae, the brown macroalgae, and the red macroalgae. The number of Thraustochytrid cells obtained from each of those was enumerated, and you could see that during the month of August to December, this bar indicates the brown algae, the green indicates for green algae, and red for the red one. Now in this, the dark brown indicates the number of brown algae which were present, but they did not have Thraustochytrids associated with them. Thraustochytrids could be isolated only for this from this many number of the brown algae. Same here, you can see that out of all these 5 types of green algae found, Thraustochytrids could be isolated from only one type of green algae and not from the other 4 types. So this variation I could see along during this study, and this was done over a period of 4 years from 2015 onwards to February. And I could see that this here for each kind of algae, for example, Padina, which is the brown algae, Hypnea, which is green, Gracilaria, Sargassum, Moldbug, all these types, I tried to enumerate the Thraustochytrid cells which were seen attached to the pine pollen when this particular macroalgae were incubated. And the number of cells, the red line that you see here, is the number of Thraustochytrids which were found to be associated with each pollen grain. So, to summarize this study of association of Thraustochytrids with macroalgae, during monsoon, lot of green macroalgae were seen, but Thraustochytrids could not be isolated, except for from one species, Ulva, during September 2018. In the season of monsoon, we could not see brown algae in this shore. Only 25% of the green algae recovered Thraustochytrids during other seasons. A higher percentage of red algae than that of green algae were positive for Thraustochytrids. And as compared to green and red algae, the brown algae had a greater incidence of Thraustochytrids. So the brown algae consistently showed the presence of Thraustochytrids in each season, though not all the species. Now, seasonal occurrence of Thraustochytrids with macroalgae was seen. Macroalgae, where the seasonal variation was seen in macroalgae, and so in the Thraustochytrids. Now, no seasonal occurrence of Thraustochytrids was found in the same region in the seawater. So, coming to that ecological role of Thraustochytrids, so the ecological implications of the associations of these Thraustochytrids with the algae. So, what are they? Now, we could see that Thraustochytrids are associated with the living macroalgae. They are also there with the drifting macroalgae. Then there is greater species richness of the macroalgae and higher percentage of occurrence of Thraustochytrids along with this macroalgae. Then coming to microalgae, the Thraustochytrid diatom associations were found to be dynamic. During the roller table experiments, you could see that the diatom polysaccharides were favorable for the growth of Thraustochytrids. In the presence of diatom tips, the Thraustochytrid numbers were very high as against the bacterial tips. But when the lipids or the fatty acids were extracted from these diatoms, these lipids did not favor the growth of Thraustochytrids. They were found to be inhibitory to these Thraustochytrids. So recently I had been on a trip to Arctic, wherein I tried to isolate Thraustochytrids from different substrates such as the sea water and also from the macroalgae found in Arctic. And there again, I saw that Thraustochytrids were again found in association with ulva and with brown algae also, and the studies of which are ongoing right now. So with this, I would like to end here, and I am ready to accept any questions.
Thank you, Varada, Dr. Varada Damare, for the very insightful talk on algae and Thraustochytrid association. If the ladies and gentlemen, if the audience has any questions, please do ask. Please raise your hand if you have questions. Yes, Mr. Raja, please ask your question. Okay, I think I need to unmute people. Okay, Mr. Raja, please. Mr. Raja, you can ask your question.
Very nice talk, please. Actually, I have one question related to the taxonomy of Thraustochytrids. So there is a lot of complexity in the species level identification of few genera like Orangeochytrium or Schizochytrium. So is there any molecular markers to delineate the species?
Usually, the Orangeochytrium was first grouped under Schizochytrium only. So later on, then it was completely taken out from Schizochytrium and put it as a new group, Orangeochytrium. So to identify actually the Orangeochytrium, you want to know the between the species of Orangeochytrium or from Orangeochytrium with the Schizochytrium. So usually what I do is we check for the 18S region. And using there are other spatial genes which are from or the probes which are generated by Professor Daisuke Honda from Japan. He has developed these probes for identification of this group of organisms. And usually when we do 18S studies and we use the traditional those other markers, the NS1, NS8, we can get this phylogeny, but using the markers or the probes by the Professor Daisuke, it will be more specific for this particular group of organisms. So then that would get more far easier than otherwise.
Thank you, ma'am. One more question. You have done very good work on Thraustochytrid associations with algae, right? So have you found like which genus or which species is dominant in them? Like you have studied various algae.
Yeah, actually, I have found a variety. I saw Schizochytrium associated during certain seasons, then Parietichytrium, Oblongichytrium. Now, Oblongichytrium was also a type of Schizochytrium. It was placed along with Schizochytrium earlier, then later on grouped away, though it was like similar to Schizochytrium. So actually, when we do molecular studies for identification, we also have to study the life cycle so that we can see the each and every stage of the life cycle, and we can differentiate between this genera. Now, generally, with as such, I can say that Thraustochytrium is present with the drifting algae. Then Schizochytrium, Parietichytrium, Oblongichytrium was there along with certain algae. And but these studies, I feel it's only a 4-year study, and that too with a few species only I could get the isolates. I had lost some of the isolates during subsequent subculturing. So, and therefore I'm being working. I can tell you it's not a representation of the entire number that I got. To identify all of them, all had to be living to identify them. So I could not, all of them could not be maintained actually. Only a few cells, a few cultures were maintained, which I could identify them later on.
Thank you for the detailed clarification. One more thing, during the isolation process, I used to encounter a lot of yeast contamination or bacterial contamination we can avoid. But Plastochytrids association with yeast is which is very tough. So how do you separate? If you can provide any insights, it will be useful.
Yeah, so usually what I do is when I am growing them in pine pollen, I will pick up only the pine pollen and put them on the plate. But sometimes yeast are also there, and then when you streak this on the plate, I try to cover the entire plate during my streaking so that they are spread out a lot. And I usually check every say 10 to 12 hours. I will check the plate for little growth. The yeast will grow faster, so therefore you have to pick up the Thraustochytrid before the yeast actually comes and over these Thraustochytrid cells. So, and this picking can be done by with the help of a needle. So you have to be very quick and very precise to picking up that one particular small tiny pinpoint colony, or just those 2-3 group of cells to be picked up with the needle. So picking it with the needle and then putting them again in seawater and pine pollen is a better option to separate them from the yeast.
Thank you very much.
Mr. Raja, if you have any question, you can ask now. Mr. Mohan Kumar.
Yes, madam. This is adding to the Devadatta's question, that yeast contamination. But again, it will grow very fast compared to other type of algae. So how do we point out the very easy isolation of that thing, madam? Yeast separation of.
What I said was picking up the cells is picking up under the microscope. So what I usually do is I check the growing colonies under the microscope under 10x objective. I point out whichever part I have to pick. Like now, suppose you have a colony of Thraustochytrid growing and yeast attached over here like this. I will pick up through the needle at this end only. I will not touch this part. Then only I'll be able to pick the yeast-free Thraustochytrids. Otherwise, if I try to take up with a loop, the loop may touch both the colonies at a time. So you have to pick up with the needle, and you have to be like, when we are growing them from the pine pollen onto the plate, you have to be very, every like a few hours, we have to observe the plate to see that we are getting Thraustochytrids and the yeast are not coming over them. It's a very tedious process, actually.
Okay.
Yeah, it's a tedious, ma'am. Yeah.
Yeah.
Dr. P.N. Singh, if you still have the question.
Yeah, that Dr. Damre. Hello.
Yes, sir. Yes.
Can you hear me?
Yes, we can hear you.
Yeah, as you told that these Thraustochytids are the percent association, they are associated with the majority with the brown algae rather than green algae. So what is the reason behind that?
This is something which I'm still finding out. I'm working on that right now, because I read that was very interesting. But initially, I thought that there are no Thraustochytids associated with green algae. But to my surprise, I found that ulva species, especially ulva, gave me Thraustochytids in culture about 3 times. Twice from the intertidal shore in Goa, and one from the Arctic. So there must be something which is really intriguing, and I'm finding that I'm working on that right now. Right now, I don't have an answer for you. This work is ongoing.
Another simple question that regarding this isolation. So are you not using that serial dilution, that because that whatever the association of that zooplankton or phytoplankton or other yeast group, so that the serial dilution is not appropriate technique to separate that pure colony or pure cells of these Thraustochytids?
Do you mean serial dilution of the pine pollen incubation, or serial dilution
No,
of
whatever.
the specimen?
I mean that purification of to get that pure cells of these Thraustochytids.
Serial
Because
dilution can be done, but again, the thing is, by doing serial dilution, we have to observe under the microscope that you are getting only Thraustochytrids attached to the pine pollen. But that will be very difficult to see if there is one cell of yeast attached to the pine pollen along with Thraustochytrid. So to understand that, we need to stick it on the agar plate only. So serial dilution can be a method, but finally it is again sticking on agar plate. So whether you do serial dilution or you don't do the serial dilution, you have to stick on agar plate to see that there are separate Thraustochytrid colonies growing or yeast attached to them or not.
okay, okay. Thank you very much, madam. Yeah.
Any last questions? Last question? Anyone? If anyone has any question? Dr. Shruti Talashi, please. Shruti, you can ask a question if you have.
Can you hear me now? Good afternoon, Dr.
Yeah.
Damare. Hello.
Yeah.
I have a question since there has been a lot of technical talk. I would like to know the applied research part of your research. Like, where do you see your species and what magical thing they are going to do for the future of biotechnology? So, can you please give some insights on it? Thank you.
So this talk was mostly, my work so far was mostly, it was all about ecological role, how they are associated with different groups of organisms. I'm not yet ventured into the biotechnological aspect. So under biotechnology, I'll be looking at the types of pufas which are produced by these organisms. I know that these produce DHA, but what concentration of DHA and whether it can be exploited commercially, and that can be done by optimization. So this is one thing I want to look into future. And also when I check the types of pufas which these types of fatty acids which these organisms produce, if there are large amounts of saturated fatty acids in some of the cultures, then this can also be exploited for biodiesel purpose, which I had mentioned earlier. So this is another thing that is the second aspect which I'll be looking for later on. But the first aspect is the pufas, the DHA part.
Thank you so much, ma'am. Thank you.
Last question, Dr. Bodhidharma, please. Okay, Dr. Rahul, Rohit Sharma, final comments from you, please.
Yeah, thank you, Dr. Shnoy. Thank you, Dr. Damre, for a very nice, informative talk.
Thank you very much to all of you. Thank you to the organizers.
The number of questions that we encountered for the isolation purpose itself shows that how difficult the group of organisms is to work on it. So it was a very nice talk, and good to know their diverse habitat that they harbor, and also their osmotrophic nutrition. And your slides also was very nice to see that the association of their macroalgae, the Thraustochytrids, as well as their seasonal variation, and how they are having a different association with the, as Dr. Singh has asked, that about the brown algae and the green algae. So very, very nice having kind of thing. And thank you so much for joining with us and delivering your talk on the International Seminar on Marine Fungi. Thank you so much.
Thank you very much. Thank you to the organizers for giving me this opportunity. And I'm really grateful to all of you. Thank you.
Thank you, Dr. Varada. Thank you so much. Ladies and gentlemen, now we'll move on to the next talk by Dr. Manjusha. She's already here. Dr. Rohit Sharma, please.
Yeah. So Dr. Manjusha is a microbiologist with a PhD from Cochin University, and she has over 15 years of teaching experience. She began as a guest faculty at Union Christian College and has since worked in various academic roles. Currently, she is an assistant professor at Kerala University of Ocean Studies and Technology and serves as a course coordinator of marine microbiology. Dr. Manjusha has completed major research projects and has extensive publications in her field. Her research focuses on marine microbial diversity, particularly mangrove yeast, and she has contributed significantly to the study of biotic potentials. We welcome you, Dr. Manjusha. The floor is all yours. Please go ahead with your talk.
Thank you, sir. Actually, shared my screen.